Hello, everyone. And yes, as Amanda indicated, I am not, in fact, Jamie Wagner. I'm the last minute understudy, but I am the lead brain tumor regulatory support in the City of Hope's T-cell therapeutics lab. And prior to joining TCTRL, I also served 5 years in safety and data quality, specifically in the Office of Data and Safety Monitoring, specifically supporting the Data and Safety Monitoring Committee. So as Jamie indicated, I also have no conflict of interest to disclose. So the purpose of this presentation will be to define the types of deviations, how they affect clinical trials, and specifically your operations as nurses. Talk about a few examples, and then also talk about corrective and preventative actions in order to try to mitigate the deviations. So what is a deviation? A deviation is any change, divergence, or departure from the protocol. And these can be either anticipated or learned after the fact. So a handful of examples include patients that are missing a visit due to traveling or being out of the area, using an expired consent form to obtain consent, and then also enrolling participants who do not meet eligibility criteria. Maybe a dose modification that's not per protocol, maybe failure to report an SAE in the timeline that's specified in the protocol. So generally speaking, deviations should be avoided when possible, but we all know that they happen. And so we'll talk about a few ways that we can try to avoid them. So in terms of deviations, there are two types. So we have unplanned and planned. So unplanned deviations, they occur and are identified after the fact. So an example would be, let's say you're in clinic and patient is consenting, and then the next day you go to scan the consent forms and you realize that patient signed on a different line or didn't put the date of the signature. Another example would be, let's say, despite the fact that the orders were placed by the CRN, if a urinalysis was not completed at a visit, then that would be considered a deviation. And you don't necessarily find out about it until the patient's already gone and you can't, collect that sample. The other type of deviation is a planned protocol deviation. So the key word here is that these are identified before, and then when we request permission from the IRB. So the both events are recorded in terms of City of Hope, they're recorded in Encore, but a PPD, a planned protocol deviation, those go specifically to the IRB, and then they also require prior PI approval. And so the way that we obtain that is on the form, they're routed to the PIs. And so their signature of that form is their attestation that they approve, that they plan to proceed with this deviation and they approve of the deviation. And then prior IRB approval is required before implementation. So again, both of these types of deviations require reporting in Encore. But there is a subset of deviations that require expedited reporting in our regulatory system, being IRIS. So generally speaking, the distinction between reporting and non-reporting, or not reporting in an expeditious manner, would be a question of severity. So does the deviation alter the risk-benefit ratio of the patients? So in terms of the unplanned deviations, there are three types within the City of Hope policy. So we have minor, major, and critical. So minor deviations are any kind of divergence from what is written in the protocol, but they don't rise to the level of majors. Major deviations, on the other hand, are those events that impact patient safety and data integrity. And then critical deviations, which we see infrequently, would be the findings that have some kind of suspected fraudulent activity or some kind of deception. So when these are reported, so the minors are reported in batches, and they're reviewed in a summary form by the committees, whereas majors are reported expeditiously and are and this assessment is done by not only the PI, but also the DSMC. So looking at these events through a different lens, the IRB, when they receive these events, they also need to assess for whether or not the event meets criteria for an unanticipated problem. So by definition, a deviation is unexpected because we are deviating from the protocol. We don't expect to do that. Also, by definition, it's related to research because whatever is not happening or whatever is happening is a protocol-driven procedure. And so when there's some kind of deviation from that, it is by definition related to research. So ultimately, the question becomes, did it affect patient safety or data integrity in such a way that would place the patient or the study at a greater risk of harm? And so, and it comes back to that same question that's being assessed when majors are assessed by the DSMC. So ultimately deviations happen and the goal is to try to mitigate them. And then when they do happen, to report them in the timeframes that are required by your institution. So when a deviation does happen, what do we do? So as part of the reporting process, after the fact, after it occurs, there's a two-prong approach. So we have to come up with a corrective action and a preventative action. So the corrective action it really gets to how did this happen? Why did this happen? We ask that the study teams conduct a root cause analysis to determine where was the breakdown, what happened, and then on the other side of the coin is to determine what can we do in the future to try to prevent further deviations of this type. So referring back to the example we mentioned, so in terms of the urinalysis that was missed at the day 14, so it was determined that the kits did not include the specimen cup. So that would be the root cause of the event, that the UA was not obtained, is despite the fact that the orders were placed because the kits were incomplete. So then in terms of the preventative action, and mind you, these need to be documented in either Encore or IRIS or whatever system is the reporting system to the committees. So then the preventative action, we would then propose that perhaps the instructions would be reviewed within the lab manual to ensure that they're complete, or perhaps a checklist be created for the biospecimen coordinators, or retraining just to ensure that it doesn't happen in the future. And these tend to be concrete steps that we need to take. It's not just this general statement of, staff will be retrained. Like there needs to be some kind of action taken within the explanation. So in terms of deviations, how do these affect you and how do these affect your daily operations? And then how can you mitigate them? So in terms of your role, you can help mitigate deviations by informing the patients of the importance of sticking to the treatment plan and sticking to the protocol. So nothing, as Jennifer mentioned, nothing is a surprise. And so they can plan accordingly, which will ensure compliance. Also rescheduling patient visits. So it's always better to reschedule and then report an out of window deviation as opposed to a missed deviation. So when you can, rescheduling events is helpful. Also retraining staff and keeping up with training, and then also the providing guidance on strengthening and optimizing the protocol throughout the life of the protocol. So we know that it's stressful. Deviations are stressful. They're stressful for the patients. They're stressful for the clinic staff, the nursing staff, the regulatory, all of the support staff. So the best approach to deviations is to try to get ahead of them. And so how can we do that? So we can try to avoid, and this really speaks to the preventative component of that CAPA. So it includes protocol development, training, and optimization. So protocol development, the important part here is that we really try to get the protocols right from the start. And I'll get into that a bit more in a minute. Also training, so attending the SIV, keeping up with the protocol trainings and the amendments that come along, and then also protocol optimization. So in terms of noting what is not working and working with the protocol writers and the regulatory specialists in order to submit amendments when there's a breakdown and we need to try to mitigate deviations. So in terms of protocol development, protocols should be written to start. They should be written with sufficient flexibility to avoid deviations. So sometimes we don't know what we don't know, and so it's an ongoing effort, but ultimately a lot of effort should be put initially to try to get things situated ahead of time. So this involves nursing coming in and reviewing the documents, reviewing that X page that Stacey noted, the study calendar, the eligibility criteria, just to account for, maybe in terms of a some kind of parameter, we need to include an allowable range for a lab value as opposed to an actual, value, or visit windows as opposed to, saying day 14, well, maybe it can be day 14 plus or minus 2 days, just to allow for there to be flexibility. Because please note that if it says day 14, it has to be on day 14 if there's no window, which, things happen, cars break down, kids get sick, and patients may need to come in a day early, a day late. And so having that window will allow for that flexibility. Also, if you pull from your nursing experience, you'll really know, because you're the boots on the ground, you'll know what is the strain for these patients. So the multiple visits per week or the time of day or whatever, you'll really be able to see what is not working. And so when we build out protocols ongoing, you'll be able to take that experience and then point out any problems early. Okay. So in terms of the training, so if we get back to how do you as nurses avoid deviations? So ongoing, we have training. And so one way is to either create schemas or reference the schemas that are in the protocols, just to kind of compartmentalize the different phases of the studies, and whether it be for the patient's understanding or for the support staff, but just to compartmentalize. We have the screening phase, which may include screening, leukapheresis, manufacturing, eligibility review. Then we have the treatment phase, which may be active treatment, and also DLT evaluation period. And then also follow-up, which depending on the study could be short-term follow-up, it could be long-term follow-up for 15 years. So just to set those expectations and create that visual tool to be able to educate patients and staff. So also this is another example. So creating visual workflows. So in this case, this is the infusion dose preparation workflow. And so this allows staff to see what is happening when, who's responsible for what, and just to make sure that steps are not skipped throughout the process, to ensure that, we are mitigating deviations. And so the last component of avoiding deviations comes down to optimization. So, when we write these protocols, we do our best to try to anticipate, especially if it's a new disease indication or a new type of study, we do our best to try to anticipate how they're going to go, and based on, previous knowledge and whatnot. But when it comes down to it, the optimization of the protocol really happens when we're doing it, when the studies are rolling. And so one way to really try to correct any kind of insufficiencies that are within the protocol is we call on the nurses to tell us what is not working. And so one way to do this is to get feedback about the protocol and just like the daily operations, because the people that are amending the protocol don't necessarily know how things are going. And if something is frustrating for you, we won't necessarily know. So it's important to communicate those insufficiencies. So an example that I have, so one of our studies, we had a pre-lymphodepletion CSF collection. That was a standalone appointment that was, it required MD time, it required CRN time, and it was difficult to, the CRNs were doing it, the PIs were doing it, but it was difficult for the patients. And so our wonderful Julie Kilpatrick, she approached us and she asked that, she proposed that appointment, that CSF collection, be added on to the surgery time point that was right before the pre-LD CSF pull from the Rickham catheter. And so we were able to update the protocol and move that collection. So we were still getting that baseline CSF collection. But it allowed us to optimize the timing and just to reduce burden on the clinic staff and the patient. So I cannot overstate how important it is for CRNs to provide feedback, whether it be in the beginning when we're writing protocols or just throughout the protocols, in order to really optimize the protocol operations, but also just the clinical trial in general. And then in order to avoid deviations moving forward. And that is it.